Extracellular vesicles, or EVs as we call them, are tiny packets of biological materials ranging from 20 to a few hundred nanometers. They contain nucleic acids, proteins, and lipids, and their role is uncertain, but everyone has a theory. But it's really like the Wild West in the research world because measuring them is really difficult. Exosomes are produced via the endosomal pathway. They're made inside the cell and then released. They can remain in the bloodstream or other fluids such as CSF, joint fluids, or urine, and they can be taken up by other cells, transferring whatever is in the EV to that other cell. There are theories about the role of EVs for just about everything, but there is little doubt now that EVs are involved in many pathologies. For example, EVs have a major impact in cardiac disease, where it is well known that endothelial cells, which line the blood vessels, when injured, are a key element in the complex pathophysiology of atherosclerosis and triggers the release of endothelial cell-derived EVs. There are numerous other examples of the impact of EVs. For example, there is now clear evidence that EVs are important in the neurophysiology of AIDS as well as in drugs of abuse, where it has been shown that numerous EVs can cross the blood-brain barrier and release their carrier molecules, resulting in neurotransmitter release. So how do we know EVs are even present in a fluid, and how do we find them? Well, there are literally thousands of papers claiming to be able to measure EVs and hundreds of techniques developed for catching these tiny packets of biological material. For example, this method uses a small chamber that removes large particles and allows the tiny EVs to be transferred to another chamber. But the most common techniques for measuring EVs are flow cytometry, nanoparticle tracking, resistive pulse sensing, and dynamic light scattering. Flow cytometry is probably the most common technique, but one thing is clear, as noted in this recent paper, and that is, quote, "Currently, there's not one single method which allows phenotyping, sizing, and enumerating the whole range of EVs, and therefore providing all the necessary information to truly understand the biology of these particles." A lot of the studies are published using flow cytometry, but here is the fundamental problem with flow cytometry. It's about the size of the particles. Flow cytometry was designed to be really good at evaluating blood cell-sized particles, but when you get to EVs, it's simply not capable of making useful measurements. But that has not stopped a huge number of publications, with the majority using a comparison technique with small beads to calibrate their instruments, but the results are less than satisfactory. Even when groups make determined efforts to modify instruments and reagents to get the best possible results, it's still a very complex process to obtain data that can be verified. So given these difficulties in measuring EVs, what is the growth rate in the EV field? A paper was recently published to determine the importance of the EV field in terms of diagnostic and therapeutic impact, and the authors studied the recent published literature. This graphic shows the number of articles published each year with EVs as a key word and shows less than 50 per year in 2000 and over 1,100 in 2016. By highlighting diagnostic and therapeutic technologies and reviewing the surrogates of exosome publications in these areas, they identified over 180,000 cited keyword, exosome or microvesicle over the 16 years of review of English language articles. More than 110,000 of these citations occurred in the last 4 years of this survey. These authors also searched Scopus, and they plotted the number of articles per year as shown in this figure. In my own survey of the literature, I looked at all papers published in PubMed since 2000 using the keyword extracellular vesicles, and I noted that 93% of all publications have occurred since 2014. The previous authors also searched the USPTO database and found that since 2000, a total of 524 US patents cited exosomes or related names, with a sharp increase since 2012. There was an interesting correlation between those patents linking those same terms to measurement techniques like flow cytometry, NTA, or ELISA. And if you follow the money, you can see a dramatic increase in funding by NIH from 2015 as the community faces the reality of new opportunities. So what is the therapeutic opportunity here? Coming back to the recently published survey, the authors note that currently there are 35 clinical trials underway relating cancer to exosomes. Approximately 2/3 of those 35 trials relate to diagnostics and the rest to therapeutics. They also identified that the global market of exosome diagnostics and therapeutics is projected to grow from $16 million in 2016 to $111 million in 2021, a 5-year compound annual growth rate of 47%. And they identified that the major factors contributing to this growth include increased demand for the use of exosomes as biomarkers in the diagnosis of cancer. But what if a new technology enters the marketplace that can easily, quickly, and accurately provide a low-cost alternative to current inadequate measurement tools? MyBlue enters the field. The answer is in the Blu-ray disc we use for playing high-resolution movies. The technology embedded within the Blu-ray disc is so advanced that it can resolve very small signals, and it is already mass-produced, so it is an ideal technology to modify for a diagnostic solution for EVs. MyBlue was the result of some serious scientists and engineers looking at the problem and finding a solution that was literally sitting in almost 1 billion homes around the world. These scientists looked at how the Blu-ray technology worked and came up with a solution that changed it from playing music and movies to reading tiny particles, just like the pits are read in a regular Blu-ray disc. And this disc contained tiny microchannels so that fluids could be placed inside the disc. They used the Blu-ray read head to read the disc just like the regular Blu-ray player, but their Blu-ray player read what was in the liquid in those channels and reported on the size and number of the tiny particles. By creating a variety of discs, they developed a new technology they called reflective phase grading. It's complicated, but using the Blu-ray laser, they created a technology that uses several signals that, when combined, allow you to measure the size and presence of those tiny particles. They could even calculate the intensity of the signals using modeling tools to design the best possible disc format. And they confirmed their measurements by using electron microscopy to see both the special grooves they created on the discs and the tiny particles. What they created was MyBlue, an alternative technology for measuring EVs. To take this to the marketplace, they created a new company called Celtient. The MyBlue technology fits perfectly into the largest area of growth predicted by the 2015 Frost and Sullivan report on the field of flow cytometry, the growth area of microfluidics with the potential for point-of-care application. MyBlue has been well documented in the research and development laboratory, where the engineering team has integrated all of the components to show a fully operational technology. There are 4 key technology developments that define the MyBlue technology, and these are the developments of the disc itself, the disc reader, and the associated electronics, and most importantly, a new single photon detector that is required to make the very small measurements from EVs. There are many market opportunities for a technology that can measure nanoparticles, including biomedical such as blood, CSF, joint fluids, and urine, a significant need in the research laboratory for cell culture-derived EVs, and a growing industrial field as so many products now contain nanoparticles. We now know that microplastics have been identified as being a potential threat. We know that the distribution of plastics in the ocean is enormous. The National Oceanic and Atmospheric Administration, NOAA, points to a huge issue of pollution by plastics. Many organizations are attempting to mitigate the proliferation of plastics in the ocean, but it may be the microplastics from nanoparticle size to microparticle size that are the most dangerous to humans. Microplastics gain entry to our food supply in numerous ways, and while we may be able to find millimeter-sized particles and even filter them out, there are virtually no filters that will remove 50 or 100 nanometer-sized particles so we consume them, from bottom feeders all the way up the food chain. Microplastics are now considered to be a threat that we must pay attention to. This requires new, easy-to-use, accurate detection and analysis technologies that can focus on the nano domain. Current technologies are simply not adequate to solve this problem. Technologies like electron microscopy have the resolution, but are expensive and time-consuming and impractical to use. A variety of nanomeasuring technologies are available, some using light scatter, others using nanoparticle tracking, Raman imaging, and flow cytometry. And while they all work to some extent, none are easy to use for nanoparticle analysis or low cost. What is needed is a simple technology that can take a liquid sample and measure the size, number, and fluorescence of a nanoparticle. That technology is what we're currently developing and which we believe answers the need for nanoparticle analysis.